Good evening, ladies and gentlemen. I warmly welcome you all to the 7th Pre-Conference Webinar of Peradeniya University International Research Symposium 2021. iPURSE, the signature annual conference of University of Peradeniya, is scheduled to be held on 11th and 12th of November 2021 at the Faculty of Arts, University of Peradeniya. It promises to be a wonderful platform for researchers from a wide spectrum of disciplinary backgrounds to showcase their research output. The webinar is a part of a series of pre-conference webinars organized by the iPURSE committee together with the International Relations Office of the University of Peradeniya. Today's webinar is organized by the iPURSE Committee of the Faculty of Veterinary Medicine and Animal Science and is titled Emerging Transboundary Animal and Zoonotic Viral Diseases, and it's going to be presented by two eminent personalities in the field. Without much ado, let me invite the moderator, Dr. Kalupahana, veterinary virologist and the head of the Department of Veterinary Pathobiology, Faculty of Veterinary Medicine and Animal Science, to start the proceedings. Dr. Kalupahana, over to you, sir.
Thank you, Dr. Sampath, for that kind introduction. It's a pleasure to be here and to moderate this session. Very good evening to participants from Sri Lanka. Good morning and good afternoon, whichever applies to the participants from overseas. Let me welcome all of you to the iPURSE 7th pre-conference webinar on emerging transboundary animal and zoonotic viral diseases and its impact on animal and public health. So today we have two eminently qualified speakers, Dr. Aruna Ambagala and Dr. Hemali Kothalawala. And the two topics that we are dealing today are Transboundary animal and zoonotic viral diseases by Dr. Aruna Ambagala and socio-economic impact of transboundary animal disease by Dr. Hemali Kothalawala. So before I introduce the first speaker, let me go through some of the ground rules of this webinar. We do not encourage you to raise hand and ask questions verbally. Sorry about that, because it may disturb the presenter as well as the flow of the presentation. Instead, I invite you to use the chat box to post your questions. Also, please make sure that you write the name of the presenter with your question so that I can direct your question to the relevant presenter. We have dedicated ample time to answer your questions at the latter part of the session once both presenters complete their respective presentation. Let me introduce the first speaker, Dr. Aruna Ambagala. Dr. Aruna Ambagala is the section head of Mammalian Disease Unit at the Canadian Food Inspection Agency, National Centre for Foreign Animal Disease, Winnipeg, Canada. Dr. Ambagala received his Bachelor of Veterinary Science degree from Sri Lanka in 1994 and his PhD degree in Veterinary Virology from the University of Nebraska-Lincoln in 2003. He completed his postdoctoral training in molecular virology at the National Institute of Health, Maryland, USA, and at HIV laboratory at the University of Toronto, Canada. He joined the Canadian Food Inspection Agency, Lethbridge Laboratory in 2010 as a research scientist. Then in 2015, he moved to the National Centre for Foreign Animal Disease of Canada, located in Winnipeg, as the section head at the Mammalian Disease Unit. Dr. Ambagala is an adjunct professor at the Faculty of Veterinary Medicine, University of Calgary, and also at the Department of Animal Science, University of Manitoba. Dr. Ambagala is the World Organization of Animal Health designated classical swine fever expert at the OIE Reference Laboratory for Classical Swine Fever at the National Centre for Foreign Animal Disease. He is working on a number of research projects on developing and validating laboratory and field deployable diagnostics and use of alternative sample types for detection of high consequence viral pathogens such as African swine fever, classical swine fever, foot and mouth disease, blue tongue virus, and COVID-19 and Ebola viruses. He also leads a number of international research projects funded by Global Affairs of Canada and OIE to enhance diagnostic capabilities in developing countries in Asia, Africa, and South America. Dr. Ambagala, today we really appreciate your time and effort in making this presentation. Once again, his topic is transboundary animal and zoonotic viral diseases. So, Dr. Ambagala, over to you for your presentation.
Before I start, I want to thank the iPURSE committee at the Faculty of Veterinary Medicine and the University of Peradeniya, and everybody, and Dr. Monasinghe, who invited me to give the talk. This is a privilege to me. It's a rare event where two of the batchmates are presenting, and the moderator is from my class, and it is a rare event. So I really cherish this moment, and at the same time, I'm nervous because I think I'm talking to my classmates and the faculty maybe after 20 years. So it's a great moment for me. And again, thank you very much. So as Anil pointed out, the title of my presentation is Transboundary Animal and Viral Diseases. Trying to move the slides. Okay, so this is the outline of my talk. I'm going to give a small introduction to CFI and CFAD. I know Anil gave a very good background of what I do, but I thought I will give a little bit of introduction of what is the role and what we do at the NCFAD in Canada related to the foreign animal diseases or the transboundary animal diseases. And then I'll talk about the transboundary animal diseases, give a little bit of introduction, and why this is a problem in the world. And then I will touch on few pathogens, which are the top on the list reported over the past couple of years by the OIE. And then finally, I will close with what we can do. And, like we have these transboundary diseases, but then what we can do to prevent and minimize the risks and the losses that are associated with these transboundary diseases. So our laboratory is part of the Canadian Food Inspection Agency. It belongs to the Ministry of Agriculture Canada. It's a government laboratory. So the Canadian Food Inspection Agency has a number of different arms. We have the operations branch, we have the policy and programs branch, communication and public affairs branch, and also legal services branch and the science branch. And we belong to the science branch, which is responsible for laboratories conducting testing and related activities used by the CFIA to verify that the national and international standards for food safety, animal health, and plant protection is ensured. So we oversee, as the Canadian Food Inspection Agency, the plants, the animal health, and also food safety. And we belong to, our labs belongs to the animal health branch. So if you look at the number of labs across Canada, we have about 13 labs, and we have expertise in all those three different areas: animal health and plant health and food safety. And we have one laboratory that works on foreign animal disease. That's the laboratory, the National Center for Foreign Animal Disease that I work in, that is located in Winnipeg. You can see the laboratories that are distributed across Canada. And some of them are food labs, some of them are plant labs, and some of them are animal labs. Our laboratory is situated in the building, it is called the Canadian Science Centre for Human and Animal Health. So this was built on the basis of One Health. So we have one side of the building, we have the Public Health Agency of Canada, which deals with the human diseases. And on the other side, we have the National Center for Foreign Animal Disease. We deals with the human disease, animal diseases. And there's quite a bit of integration, as you can see, under One Health concept. And we tend to, as the time goes, I know previously we thought, okay, there are human diseases, these are animal diseases, but as we go, as we learn more and more, we realize that they have to be studied together, work together, and controlled together, because most of the human diseases started as 75% of them as animal diseases and transmitted to humans. This building was commissioned in 1998, and by having co-localized in the same building, we share a lot of resources, the knowledge when it comes to controlling these diseases. The NCFAD particularly is, it's a very unique building. We have contained level 3 and biosafety level 4 facilities for both small animals and large animal experiments. We can house large animals in biosafety level 3 and 4 conditions. And we are the Canadian National Reference Laboratory for Foreign Animal Diseases, the only lab in Canada. And there's in the US, there's another lab, probably you heard about the Plum Island lab, which is similar to our laboratory. And we house 3 OIE reference laboratories. One is for classical swine fever, the other one is for highly pathogenic avian influenza and for foot and mouth disease. And also we house 2 FAO reference centers, one for foot and mouth disease and the other one for zoonotic and emerging pathogens. Those viruses including Nipah, like zoonotic diseases like Nipah, Hendra, Ebola, and now COVID-19. And the primary mission of our laboratory is to provide rapid and accurate diagnostics of foreign animal diseases for the CFIA National Animal Health Program. And the additional functions that we perform are technology development and research. So we work on those pathogens, foreign animal disease pathogens, and try to develop new technology to identify. We try to learn pathogenesis of these diseases, and we do also do vaccine experiments. We are the regulator for vaccines in Canada, Canadian Food Inspection Agency. So we cannot develop vaccines, but we can assess the vaccines somebody else developed. So we do participate in vaccine development and assessing those ones developed by other agencies or companies. And also we do train veterinarian scientists and students. We have a vet course every year in January and February to train the veterinarians in Canada on foreign animal diseases. So we bring in 25 to 30 veterinarians every year, and we have animals infected with all these diseases, ASF, CSF, FMD, avian influenza, Newcastle, so they can see what are the diseases from the beginning to the end, and then we do postmortems and laboratory diagnostics and show them how we go through the diagnosis, how to recognize these diseases if it happens in the fields in Canada. When you look at the building, we have about 25% of the building is CL2 and 70% is CL3. The majority is containment level 3. And also we have 3+ for zoonotic pathogens and also level 4, about 5%. And we are accredited by the Standard Council of Canada to ISO 17025, both for diagnostics and research. We have about 90 people working in. And as I mentioned before, we have multiple units: avian diseases unit and mammalian diseases unit that I work with and work in. And then vesicular disease unit deals with foot and mouth diseases and other diseases that cause vesicles in animals, like vesicular stomatitis. And then we have a special pathogen unit that deals with zoonotic diseases like Ebola, Nipah, and COVID-19. And we have a serology unit. It's a CL2, containment level 2, which does most of the serology screening. And if they find something, all the samples will come into the level 3 units, starting from the 1 to 4. And then we have supporting units for these main units. The genomics unit, which has genomic sequencing capacity, including whole genome sequencing. And then we have a reagent development unit that will provide us cell cultures and viruses, whatever we need, not the viruses, the cell cultures and any other reagents, monoclonal antibodies. And then we have the animal care unit that supports us for our animal experiments. And then, of course, we have a QA unit. And we also house the main office of the Canadian Animal Health Surveillance Network. This is distributed across Canada. This includes the provincial labs, the university labs that are participating in this network. And as you could imagine, we are dealing with a number of diseases that are not present in Canada. And you can see on the left side top, avian influenza, Newcastle disease, and a number of diseases. That's the avian units. And the middle is the vesicular disease unit. And the bottom is the other pathogens that are dealt by the special pathogen unit. And my unit, the mammalian disease unit, we deals with a number of other mammalian diseases, starting from blue tongue to lumpy skin diseases and African swine fever. Lately has been a big issue, and probably it's been major focus in our unit. Okay, so that is a brief introduction to the NCFAD, and let's move on to talk about transboundary animal diseases. So transboundary animal diseases are highly contagious and transmissible epidemic diseases of livestock which have the capability for rapid spread to new areas and regions, regardless of national borders, and have serious socioeconomic consequences. And if you look at the livestock production in the world, and you can see almost 1 billion poor people in rural areas do livestock. They depend on the livestock because there's no formal education needed to raise some cows or chicken, pigs in your backyard, and you don't need a large capital, and you don't need, usually people can get away with land ownership, so because they send the animals to the neighboring jungle or neighboring areas to grass, for food. So this is a large amount of people, and they provide more than 70% of the calories to their communities. And major transboundary animal diseases will have a severe effect on these people, livelihood of the livestock, these livestock farmers, and deprived from nutrients. Not only the morbidity and mortality that is associated with the transboundary diseases, it also hinders the communities of their ability to sell animal products such as hides, bones, wool, and also something that we don't think about is deprive the animals draft power for tilling and transport, etc. They are a serious threat to trade and travel, and therefore severely affect the national economies and political stability of the countries, and even global economy. And they can cause environmental and emotional issues due to mass euthanasia. You can imagine in countries where these diseases are not present, and when they appear in order to gain freedom, there is going to be mass euthanasia of the animals, and this can be a big emotional issue for the farmers, for the public. And also when transboundary diseases happens in wild, and there can be large die-offs in some cases, and that has a severe environmental impact. And of course, the other side of the story is the cause of pain and suffering for these affected animals due to the disease, due to the mass euthanasia. And if you look back and see what has happened previously, and you can see the FMD outbreak in the United Kingdom in 2001, it's cost over $9 billion US dollars, and over 6 million cows and sheep were killed. And in Southeast Asia, in the 2004 high-path avian influenza outbreak, H5N1, about 150 million chicken were destroyed or killed. And there was an outbreak of classical swine fever in Netherlands, which cost 2.5 billion and destroyed about 11 million pigs. And then the ongoing outbreak of African swine fever in Asia, the estimation is about $130 billion, according to the Asian Development Bank. So you can see the massive impact, the economic impact, and the impact on the number of animals, these diseases. And they are expecting, and they're worried actually, now ASF outbreak in North America and in US itself, it may cost about 50 billion over 10 years if they have to eradicate the disease if it shows up. So you can imagine the magnitude of impact of these transboundary animal diseases. How about developing countries? Developing countries actually suffers the most due to the poor veterinary services compared to the medical public health services that they have. And of course, and due to the lack of awareness of the farmers about these high threat animal diseases and lack of diagnostic facilities for exotic diseases, these diseases will show up and you don't have the diagnostic facilities to quickly diagnose and eliminate the diseases so that the disease will spread. And also due to poor and faulty compensation schemes. This is a big problem in Asia that we have seen with the African swine fever outbreak ongoing since we were directly involved in these outbreaks. This is a problem. If there is no good compensation scheme, the farmers will somehow not report the disease to the authorities, and that can create problems and the disease will spread. So you need to have a good compensation, well-planned compensation scheme. Underreporting due to fear of loss of internal and export markets, and some countries do that. They do not report to the OIE when they have. So all these things will lead to finally large economic losses, and also finally the disease will end up as an endemic disease in these countries. So if you look at transboundary animal diseases, you think, like with everything happening, with the technology, with the knowledge that we have gained over years, that these will go down. But no, what has happened is the TADs have increased over the years, and it is likely to grow further in the future. Why? There are a number of drivers for TADs, and one of the major ones is the intensive animal production systems, which is getting worse and worse, actually. This is to meet the rising demand for animal protein. So if you look at on the left side on the top, the demand for animal proteins in the world, you can see there's a huge spike that's after late 1900s, you can see the world population grow, and with that, the number, the need for the meat have increased. And it is expected to have a world population of 9.1 billion in 2025. And the food production needs to be increased by 70% between 2005 and 2050 to meet the requirement. And also, the high income of people in developing countries now generate more demand for animal proteins. We need to eat meat. We need to have eggs with each meal. And so all these things, these are good things, but at the same time, it put pressure on the production of meat and meat products. And also we want more protein for a cheaper price, so you have to intensify the production systems, and that lead to overcrowding of the animals. And often peri-urban areas under suboptimal husbandry practices are used to meet this demand. And if you look at industrialized countries, and you can see like poultry laying farms, so there are about 150,000 birds per house, and you will have dozens of them in one site. So that's highly concentrated, susceptible group of animals. And also most of the times they have not seen the disease. They're not immune to the disease, because most of the developed countries, they don't vaccinate these birds because they want to maintain freedom of these diseases. And the ideal situation for a pathogen to enter, and you have these large groups of naive animals, they have good biosecurity, but you need to be only one time correct, right? When you want to enter into these big barns, and they somehow get, if they get in, then you have the ideal conditions for the virus to replicate and cause a huge impact, and even to mutate the virus, so the virus can mutate into a higher virulence. So you can see that it happens in chicken farms, in feedlots, in US, in North America, and also now in China. To meet the demand for the meat, they are building these high-rise buildings, and they're not apartment complexes. They are pig pens, and they have thousands of pigs in these pens. They are nice when they are working well, but if a pathogen enters, that's the best place for a pathogen to propagate. What are the other drivers for TADS? And impact of changes in forest ecology. Again, driven by us, humans. We want more and more farmland, so we go and clean out the forests and bring in the farm animals. And you are introducing these farm animals that were never seen some of these diseases in wild animals. And so this way you have new diseases that are jumping from wild animals to the farm animals. And one of the best examples for this is the 1998-99 Nipah virus outbreak in pig farms in Malaysia from fruit bats. The other drivers for TADS, the one is the globalization and trade. And you can imagine that global trade is so important for everybody. It provides great quantity, variety, better quality of food for a lower price. For me in Canada, I can have a mango, I can have a banana, which never grows in Canada. Here's global trade. I can have access to a lot of good food from home. And about 25% of world food production is traded globally. So you can imagine 1/4 of the production is traded globally. What this does is, though, if you are not careful, it will facilitate transfer of infectious agents. And also, if you look at the efficiency of transport, you can reach any part of the world within 24 hours in a plane. You get into a plane, you're on the other side of the world within 24 hours, and it is less than incubation period of most infectious diseases. So an animal or a human can get infected with something, no clinical signs, no evidence of the disease, but when you land in the other part of the world, and you're coming down with the disease, and you are spreading the disease to the new community. So, and also the fresh commodities and processed foods can carry pathogens to distant part of the world and countries and continents. And most of the transboundary diseases, if you look at it, they are actually spread by the humans, our actions. And the best example from recent is the ASF outbreak in Europe. And ASF, as I will go into details later, was limited to Africa for many, many years. And there was a 1957 outbreak incursion in Europe, and 2007 incursion simply because of the food from a ship or a plane. And also, I think we need to pay attention to the wildlife trade and smuggling. And you think it is not happening in large quantities, even in developed countries, and you'll be surprised how many of these the bushmeat that's coming into Canada and US through the planes. So people bring things, and people travel around the globe for business, tourism, and they carry their delicious food with them, and with that, the viruses and bacteria from the different parts of the world. One of the other drivers for TADS is the effect of climate change, global warming, and microbial evolution that you can see. And we all agree that the climate is changing in the world. And we can see, for example, we had very high temperature. Winnipeg is a place where I live. It's minus 40 degrees for 2 months, but we didn't have very cold winters last year, and we have a very warm summer, no rain this year. So for sure, things are changing. There's a global warming and rising temperatures in the Northern Hemisphere. And we have witnessed a number of diseases that are present in Africa, in Asia, are slowly moving north. And the best example is the blue tongue 8 outbreak in Netherlands in 2006. And likely by the importation of an infected zoo animals or an infected midge. Midges are the vectors for blue tongue, which cost about 32 million euros in 2007 and 120 million euros in 2007, 32 million in 2006. So, and it is still a problem, and they have new vectors now because there were some vectors before, but the virus was not there. Now the virus is there. So the vectors that were present in Europe are now picking up the virus and spreading it across. And the other example is a Rift Valley virus, which increased in East and West Africa due to climate changes. Something that we don't think will drive the TADS is the influence of increased conflict and unrest, and which happens throughout the world. Unfortunately, civil unrest, inter and intra-country conflicts. This will disrupt the enforcement of quarantine and other control measures could be due to refugees or army deployments, movements. There's no law and order, so that it is so difficult to maintain animal health and quarantine, monitoring, surveillance. Those are not happening. So this is a problem in a number of countries in Africa that it is difficult to control these transboundary animal diseases because of conflicts and unrest. So those are the main drivers for TADS, and you can understand why it is becoming a continued issue and why it will get worse in future. So if you look at the diseases that have been reported to OIE, because all these transboundary animal diseases are reportable to the OIE, the World Organization for Animal Health. And OIE has listed about 117 animal diseases or infections or infestations. And when you look at the last couple of years, the top hits were African swine fever, highly pathogenic avian influenza, foot and mouth disease, and lumpy skin disease as an emerging disease in Asia. And of course, there are major other transboundary animal diseases like PPR, that affects sheep and goat, classical swine fever, Rift Valley virus, sheep and goat pox, Newcastle disease, and RHD. So for this presentation, I thought I will focus on few diseases, the top hits, and introduce those diseases to you, those who are not familiar with these diseases. And I think Dr. Hemali Kothalawala will discuss about a little bit more about the situation in Sri Lanka, the impact of those diseases in Sri Lanka, and including some other diseases, other transboundary animal diseases. So African swine fever, it's a contagious, deadly hemorrhagic fever in pigs with a high mortality approaching 100% in domestic pigs. There's no effective vaccine available so far. There are a lot of vaccine candidates in testing, but not a vaccine commercially available. So there is no vaccine to be used. That puts make it so difficult to control. So the only way to control this disease is to slaughter the infected animals, the suspected animals. The disease is caused by a large enveloped DNA virus. It is the only DNA arbovirus that is known. Arboviruses are the viruses that are spread by insects. This has about 24 genotypes based on the partial sequence of P72 genes. It's a structural gene of the virus. And there are all these 24 genotypes are present in Africa. Outside Africa, there are 2 genotypes. Genotype 1 in Sardinia as a result of the 1957 outbreak. Sardinia is a small island, and they could never get rid of it. And genotype 2 is the virus that is responsible for ongoing outbreaks in Europe, Russia, and Asia currently. As I mentioned before, this was a disease restricted to Africa, sub-Saharan Africa, and maintained in a complex transmission cycle. I'm not going to go into details. The cycle includes ticks, warthogs, and domestic pigs. And ticks are susceptible. This is a virus, mostly believe that it is a tick virus that jumped into pigs, because the virus replicates very well in ticks into large, in their saliva to high titers, the large numbers, and they have a transovarial, transstadial transmission and transsexual transmission that happens in ticks. So this is how it is maintained in Africa. And the warthogs are asymptomatic, and they develop persistent infection. But when the virus jumps to domestic pigs, it is almost 100% lethal. This is the global distribution. As I mentioned before, it is present in Africa, where it's first found. Still a problem, still moving and killing pigs in Africa. And it is a problem now in Europe and also in Asia. The last outbreak, as I mentioned before, happened in 2007, and this was due to some swill feeding of meat products, pork products from a plane, from a ship close to a harbor. It went into domestic pigs, the backyard pigs, and then it spilled into wild pigs. And you can see the virus slowly spread across Europe. And the green dots are the wild boars. Wild boars picked up the disease, and it slowly but steadily spread across Europe. And this is 2020. You can see it's moving towards West Europe. And again, the purple color dots are the wild boars, and the commercial pig farms and the domestic farms are orange and red. You can see the wild boars is taking the virus and moving it, and of course giving it to the backyard farms, and that's how the domestic pigs getting infected. The most recent outbreak is in Germany. So it was a major concern that it is in the Western Europe, and you can see it enters through the wild boars from Poland, and now it is causing outbreaks in domestic pigs. There were 3 outbreaks reported in Germany. So it is moving. The major concern for African swine fever started when the virus entered China in August 2018. Why? Because China housed the largest number of pigs in the world, 440 million heads. So we knew that when the virus enters into China, this is going to be a disaster and this is going to spread. So exactly what happened, it quickly spread through China, and it entered Mongolia, Vietnam by, in an year, it was in Vietnam, it was in Cambodia, and now you can see that it has spread all over the Southeast Asia, and it came pretty close to Australia. It was detected in Timor-Leste, just only 650 kilometers northwest of Darwin, a small island in Australia. And also it is in India now. It is in Assam and Arunachal Pradesh. I think it moved a little bit south a little bit over the past year, and it is in Malaysia. It has gone all over the place. So this is something to watch, and it continues to spread. How it is transmitted? Mainly by orally, uncooked pork scraps, blood, bloody exudates. Tick bites only happens in Africa. We don't have the, I don't know whether in Sri Lanka may have, but not many countries have the ornithodoros Marlborough kids, the ticks. Primary site of replication is monocytes and macrophages in the tonsils and lymph nodes, and then the virus will spread. It's very complex pathogenesis. This is a huge virus. It's a DNA virus, and only partially understood. What kills the pigs is the cytokine storm, which causes hemorrhages in multiple organs, similar to Ebola in humans, and you will see blood all over. There are different forms of the disease. You can see the pro-acute form, where you don't see any clinical signs. You go into a pig pen, they're all dead, or to acute form, where you see animals develop some diseases, but mortality is very high. Those are the main forms of African swine fever, but there are some subacute forms and chronic forms are developing as the virus spread in Asia and Europe. And this is a common issue in Africa, the subacute and chronic forms. But mostly you will see is the per-acute and acute forms. And per-acute disease, as I mentioned before, like you find dead pigs when you enter. There was Nigeria, there was a huge outbreak which killed thousands of pigs, and also some farmers died of seeing their pigs dead when they enter the pens. The disease, high fever, protraction, hyperemic ears, cold to touch. These are some of the pictures that are taken at our facility. You see bloody feces, bloody diarrhea turning into, you'll see cutaneous hemorrhages. And lymph nodes. You'll see hemorrhagic lymph nodes. There's going to be a lot of effusion, bloody effusion in the peritoneum, in the pleural cavities, in the thorax, around in the epicardial sac, sometimes pericardial sac. Sorry, you'll see a lot of hemorrhages. And the lungs, you will see interstitial pneumonia. This is a heart. You can see the hemorrhage. It can be pretty bad. You will see gastrohepatic lymph nodes, hemorrhages, gallbladder edema and hemorrhages, splenomegaly. You'll see a huge spleen in this animal. This is considered as one of the key features of African swine fever, but lately what we have found out is this is not the major sign. I think major one is the hemorrhagic gastrohepatic lymph node is pretty much the major indication of indication. It's not a confirmatory, but it's an indication that probably you have African swine fever or classical swine fever, or it could be even like high virulent PURSE. So you can see hemorrhages in the kidney. Okay, so let's move on to highly pathogenic avian influenza. The second disease that I want to talk about, this is caused by a negative sense single-stranded segmented RNA virus. There are 4 types of influenza out there. Influenza A virus causes influenza in birds and some mammals, and they are the only influenza viruses known to cause global pandemics. And they are divided into subtypes based on the 2 proteins. You hear about H5 and N9, and this is based on the hemagglutinin and neuraminidase, the 2 proteins that are present on the surface of this virus. There are 18 different H types, 11 different N types, and the virus is constantly changing due to the antigenic drift and antigenic shift. Antigenic shift is mainly because this is a segmented genome, so that if one cell or one animal is infected by multiple different viruses, they can exchange these segments and come out as a totally different virus. So that's where the virus is different, and it's a new virus, and now it can cause epidemics or pandemics. Yeah, so highly pathogenic avian influenza has become a continued threat to global health and economy. And on the OIE definition, high path AI is defined as when you infect the poultry, if you see about pathogenicity index of 1.2 or more, or kills more than 70% of the 4 to 8 week old chickens, that is considered as a high pathogenic avian influenza virus. The difference between high pathogenic and low pathogenic influenza virus lies in a small sequence of the HA genome, HA gene. It is usually low pathogenic avian influenza viruses are restricted for the replication into the respiratory and intestinal organs, but due to the change in the sequence of this special site, it's a protease cleavage site, which is needed for the virus to infect the animals. The change will allow the proteases that are present in the rest of the body to be able to work on this virus and cleave and make it infectious. So then it's a systemic infection when the virus changes from low path to high path. So in low path infections, you will see respiratory signs, maybe a little bit of diarrhea because of the target replication sites. But with high path AI, because it's a generalized infection, you will see generalized infection and high mortality. And the issue with this disease is, unlike African swine fever, which is not a zoonotic disease, but this can be transmitted to humans if it is successful. So when you enter a poultry pen, this could be an indication that you have high pathogenic avian influenza. You can see all the pigs are dead, and the other clinical signs, you may be able to see the wattles, swollen, hemorrhagic, and subcutaneous hemorrhages. Neurological signs can be seen, just like in Newcastle disease. And a lot of hemorrhages you will see in multiple organs. Again, this is a systemic disease, causes hemorrhages in gastrointestinal tracts. You can see in the trachea hemorrhages in the center. Here's a lung with hemorrhages, pancreas, and GI tract, and the heart. So you will see massive hemorrhages in multiple organs in these birds. So the transmission, as the virus is shed in high concentrations in feces, and wild birds will have other main reservoir for this virus. It will be transmitted to the domestic pigs, poultry. And from there, like I mentioned, it could be zoonotic. It can infect humans, and also the pigs. The pigs are, when it comes to avian influenza, we call it a mixing vessel because it is susceptible to both avian and human avian influenza virus. And remember, I was talking about the antigenic shift, and that can happen in pigs very easily because it receives the viruses from humans as well as domestic poultry, and it can give back new viruses to the world. So what is the importance of these migratory birds, the wild birds? And they will take the virus and move it around the world, and you will see that it is moving from different parts. That's one of the reasons it's a transboundary disease, because they don't care about the boundaries, they fly. When it is winter time, they want a warmer place, so they move from North Pole, from cold climate to the warmer climates, and they come back when we have summer. So they bring back new viruses to us. And you can see the movements of these birds. They have different flyways, and you have East Asian, Australasian flyway, which goes across China, and China is There are a number of high pathogenic avian influenza viruses arose in China, and they have been distributed worldwide by this migratory bird. And also you can spread the disease from country to country by importation of infected birds and bird products, and farm to farm by movement of people, clothing, infected birds, and boots are notorious, contaminated equipments, and feed. And probably most of you remember avian influenza outbreaks over the years. Some of them are zoonotic outbreaks, some of them are only in birds. And 1980 Spanish flu, these are all caused by different strains of avian influenza. And some of them were zoonotic, some of them were not. The main concerned strains that are circulating right now are the H5N1 strain, which was reported in 1996, and it is still circulating. And it has caused a lot of outbreaks around the globe. Millions of birds have died, slaughtered to limit the spread. And it is a zoonotic disease. Not too bad, only jumps into humans, but there were no human-to-human spread, luckily. But when it happens, it's about 60% mortality when humans get infected. And the second one is H7N9, which arose in March of 2013 in China. It arose as a low pathogenic strain, but later it became a high pathogenic strain, and mortality increased, including to 100% in birds, and in humans about 50%, and it looks like it's more adaptable to human respiratory tract than H5N1, so this is a major concern. All right, so moving to the third disease, this is foot and mouth disease. It's one of the most contagious animal diseases known to humans, which has a wide host range here again. Unlike African swine fever, which is restricted to pigs, this can affect the ruminants and pigs. It is a small virus. It's a positive stranded RNA belong to the aphthovirus genus, but the virus can survive for a long time in different, in hay, in dry feces. So this can become a problem because it's an environment. Susceptible animals are cattle, domestic buffalo, sheep, goat, and pigs, and also other animals, the wild animals like African buffaloes, gazelles, warthogs, deers, llamas, and double the camels, which the double humps, not the single hump ones. And this has been reported as a zoonotic disease, but very, very rare cases and very self-limiting, limiting to fever and rash. And the issue with this disease is that there can be carriers of this disease, only ruminants, but low level of virus shedding can be found in the pharynx samples, and it depends on the type of animal, the duration. There are 7 immunologically distinct serotypes of African swine fever virus, OACHR1 South African territory, so SAT1, 2, and 3, and you can find them as pools. There are different pools in different parts, and you can see the serotype O is the most widely distributed serotype. And the serotypes are very important. The determination of the serotype is important because there is no cross protection. So you need to identify the serotype that is responsible for outbreak if you're going to use a vaccine. If you're not going to use a vaccine, then it's fine. But if you're going to use a vaccine, you need to identify, you need to know the serotype so you can use the vaccine that is targeted for that serotype. Talking about African swine fever, I was looking at the latest hits, and you can see Sri Lanka on the map. There is a new lineage of foot and mouth disease was reported in Sri Lanka, and this was reported to the OIE in the last meeting. And this virus seems to be different from the other viruses, Pan Asia 2 and India 2001 lineages, but it seems like it is similar to the contemporary outbreaks that was happening in India. So how the virus ended up in Sri Lanka, nobody knows, but it seems like it's a new lineage of virus that was reported to the OIE. Transmission, vesicular lesions on the skin, and this has so much virus in those vesicular lesions on the skin, but also virus is shed in excreta, in secreta. And all tissues during acute phases, you can find viruses, including bone marrows and lymph nodes in carcasses. Sorry about the typo here. And method of spread, movement of infected animals, movement of contaminated products, wild animals, airborne spread is possible with foot and mouth disease. Route of infection is direct contact, ingestion, inhalation, or contamination of damaged skin through the mucous membranes. Reuse of needles could spread the disease. Pathogenesis, incubation period is 2 to 4 days, sometimes 24 hours. We have seen here, based on the experiments we have done, initial replication in pharynx and skin, then viremia, and then general infection, high level of replication in the skin. That's why you find a lot of viruses and it causes blisters. And clinical signs, fever, depression, anorexia. I mean, this is a disease that most of you have probably seen. Lameness, salivation, nasal discharge, reduction in milk yield, and abortions. Animals usually recover in 2 to 3 weeks, but you can find some death in young animals due to myocarditis. And some of the pictures that were taken here, yeah, you can see the lesions clearly, and sometimes you can see mostly in the mouth, in the tongue, and then you can see sometimes not very obvious in cattle, the coronary band lesions, there is like blanched, but less severe compared to pigs. I'll show you some pictures in pigs later, and you can see, these are some experimentally inoculated animals. You can see the development of the lesions in the feet. The lesions can be on in the udder and also inside internal organs. And the disease in pigs, the tongue lesions are not very common in pigs, but you will see the feet are very severely affected to the level they cannot walk. And you can see it's pretty bad in pigs when it comes to, that's probably the first indication that you will see in pigs infected with foot and mouth disease. And sometimes you will see vesicular lesions in the snout. Interestingly, sheep are silent shedders. Goat and pork, sheep and goat, they are silent shedders. No clinical signs, but they shed a lot of virus. Okay, so this is just to compare 3 different animals. What the disease, the role for cattle is an indicator. For pigs, it's a virus amplifier because there's so much virus amplifies. And sheep we call silent shedders for the same reason I mentioned before. Because the clinical sign in cattle mostly you see salivation, and pigs lameness, and sheep probably nothing. And foot lesion severity in cattle is moderate. In pigs, as I showed before, it's very severe. And nothing probably in sheep and goat. Mouth lesions, again, cattle is very severe. In pigs, it's moderate, and sheep probably you see nothing. When it comes to persistent carriers, cattle, yes, pigs, no, and sheep as a ruminant can act as a carrier. Okay, so that's about foot and mouth disease, and let's jump to lumpy skin disease, something that was making headlines recently in Sri Lanka. This is a non-zoonotic vector-borne poxviral disease of cattle and Asian water buffaloes, characterized by fever, firm skin nodules, necrotic plaques on the mucous membranes, mastitis, and ochitis. Morbidity is 5 to 45%. Mortality is less than 10%. LSD, once introduced into naive population, though, it can lead to significant income losses for farmers due to decreased milk production, damaged hides, emaciation of animals, infertility, and abortions. Subsequent trade bans or other movements can follow following an outbreak, and that can affect the economy of the country and the industry. So again, the disease was first observed in Africa, and it slowly made its way into out of Africa, into Middle East, Southeastern Europe, Central Asia, South Asia, and China. And this table, you can see some of the countries in 2019, 2020, how the virus spread. You can see it in India. Orissa first reported cases in 2019, and it quickly spread throughout India. And the virus was sequenced, and it genetically close to a South African virus, not compared to the European isolate. So we don't know the origin of the virus yet, but there were a lot of outbreaks happening in Europe. So we don't know how the virus entered into Asia. It was observed in August 2020 in Sri Lanka, in northern parts of Sri Lanka, and quickly spread after the heavy rains. This is again showing the countries that are affected. So you can see the latest outbreak was reported in Laos in June of 2021. So just like African swine fever, it is spreading. And of course, like African swine fever coming to China, when it came to India, that was a major concern because India has the world's largest, the highest heads of cattle. 303 million cattle. So it was a concern. It spread so quickly, and you have the vectors to spread, mechanical vectors, I will mention in the next slide, and so the best place for the virus to be. Vectors include mosquitoes, biting flies, and ticks. So pretty much it's a mechanical transmission. There's no virus replication in the vector, and also a needle use, of course. Anything that can poke the skin can transmit the virus from one animal to the other. Incubation period is about 12 days. Clinical signs, fever, skin lesions, it's pretty obvious. These animals, it's hard to miss any of these animals with the LSD. Skin nodules, and you can see ocular nasal discharge, abortions, and mastitis. Hides will be completely damaged. Others are affected, leading to mastitis and eyelids, and you can see erosions in the mucosal surfaces, the lip in this animal. And also you will see the nodules in internal organs. In abomasum, the serosal surface, you will see the lesions. And the lesions in the lungs will cause pneumonia, and also sometimes you will see a lameness in these animals due to the inflammation of the tendons. And so it is a debilitating disease, and it's a disease for concern, especially to protect the dairy animals, dairy industry, and that can have very bad consequences. Okay, so the final few slides I want to talk about how we can manage, and we know that these diseases are spreading. There's nothing stopping them, it seems, but we need to have a proactive approach, and this is the only way we can stop them. And if they show up, then we can quickly eliminate or control them. This involves, I know it's been always a disease, a problem for the governments, but I think that thinking pattern is changing. I think it has to be a multidisciplinary collaborations when it comes to fighting these diseases. Public-private partnerships, international collaborations, and scientific progress is needed. And private industry has to contribute and help with the government to control these diseases, because they benefit from from animal industry. So they should contribute, and they should work with the governments. And international commitments are important. Reporting and also working with international groups, because we all know that we cannot work alone. These diseases are transboundary diseases. We need to work together as an international community to control these diseases. Those countries who doesn't have the diseases need to make sure that they help the countries who are suffering from these diseases and to eliminate those diseases, so those diseases will not come to their countries. So that should be so it has to be an international effort to control these diseases. And of course, a well-educated veterinary workforce is needed to provide scientifically sound risk assessments and credible reporting of the diseases and effective delivery of services to the producers. So when I think about it, I can come up with four things that we need to pay attention if you want to manage these stats effectively: the foresight, the prevention, early detection, and swift and effective reaction when the disease shows up. So the foresight is scanning the horizons to identify the source of pathogens, what is out there, what are the things, what are the hotspots, what are the risky practices, and this is all comes with the knowledge and knowing what is out there, and then you can come up with an emergency plan for dealing with these outbreaks if they shows up. Of course, this is always tough, even for developed countries, to invest on foresight, because we all know that it is not always possible to 100% prevent just by doing foresight these diseases. So that kinds of it's not very palatable for the governments, for the industry, but I think that is very important. The foresight is very, very important, and that is something we need to stress. Because I think this will give us the ability to respond. The prevention, it's always the best. The knowledge that you gain from the foresight exercises to develop preventive actions to reduce the outbreaks. For example, like when it is happening in India, what can we do to prevent the virus shows up in Canada? What are the biosecurity issues? Do we have to educate the farmers on this disease so they can quickly pick it up if it shows up? We have to identify the vulnerable communities and educate them. And then you have to ensure that we have safe practices. And you can see what's happening in Asia is they do not have safe practices when it comes to production, processing, transportation, and even marketing. And that helps the spread of these diseases. And if you really look at it, it's us humans are responsible for spreading these diseases across the globe. And firm control of livestock and livestock product imports. That's a key thing for countries like Sri Lanka. We have to make sure what we bring in. We have to make sure they're disease free, and so that we don't end up with the transboundary animal disease. Early detection. Early detection is critical because we know with all measures that we can take, we are going to face transboundary animal diseases or any emerging disease outbreak. So you need to have the diagnostics ready. You have to have effective veterinary services in the country with the vigilant surveillance system, and also good lab support, state-of-the-art techniques, and ability to do high throughput screening. And I think one of the most important things that we forget is the traceability. I think traceability is important because if you have a surveillance program, if you cannot trace where the animals came from, I think it is going to be useless. So traceability of animals is very, very important to go back to the farms and ensure the quarantine and take the measures. And there are tools out there developing, and this is some of the things that we have been working on: real-time tools, portable detection tools that you can take it to the field and detect it, and then immediately, this is a portable system that can work using a cell phone, and then you can quickly update to the central lab, so you can see real-time what is happening in the field. It will show up in a computer. Different parts of the country, the disease happening. So this was developed by us for African swine fever disease surveillance, and if it comes to Canada. The last one is the effective reaction. It has to be quick. You have to, once it is detected, you have to make to take action before it becomes it surpasses the critical level, because after a certain threshold, it is endemic in this country. So you need to act quickly. But to do this, you need the resources, and you have to make sure the emergency funds are available, and then you have the ability to mobilize them. It could be the money, it could be human resources, it could be the lab resources, the vaccines. You need to have them ready so that you can mobilize them. Powers to enforce meaningful control of livestock movements, and that's very, very important. You should be able to control the movements of the animals, quarantine the animals, and when you should have the complete authority, like CFI has the complete authority to stop animal movement in Canada. Compensation for losses, that's very important. I mentioned before too, because if you're going through stamping out, there's loss of trade, the income is going to be lost. And what happens in Asia is that the compensation, either it's lower than what you get from the market price for a pig, or it takes forever for those compensation to happen. So people will just not report. They will send the animals to the slaughterhouse, or they will kill in their backyard and sell the meat. So that is not a way to control and transboundary outbreak. Animal disease outbreak. So again, I think synergies between animal, human, and environmental health is important when we control transboundary outbreaks. Collaboration. I just wanted to emphasize collaboration. I think, as I mentioned before, any of these diseases, transboundary animal diseases or any emerging infectious diseases, even in human diseases, it has to be a collaboration locally, nationally, internationally, between the veterinary community and as well as the environment. Those people are responsible for the environment and also the human health. So it has to be a collaboration, and with clear delineation of roles. You need to identify your roles before, and the responsibilities of each individuals, each group, and then your boundaries. This way is the only way we can control these diseases. Creating and support of expert network to address specific issues. And we need to overcome the reservations in sharing information. And I know a lot of countries do not want to share their epidemiological information on the diseases, which is not helping in a global context for controlling these diseases. And some people don't want to share their biological information or genetic information of the viruses they have. But I don't think these restrictions should be overcome, because otherwise it will be so difficult to control the transboundary disease outbreaks, because it is not a problem for me today, but it can be my problem tomorrow. So this is the attitude we all need to develop and share the information and share the knowledge and work together to deal with the transboundary animal diseases. With that, I want to stop, and I want to emphasize that there are so many help out there for countries. GFTAD, I think Dr. Kothalawala will mention some of these again. OIE, FAO, when it comes to African swine fever, Global African Swine Fever Research Alliance, and they're willing to share the protocols, any information you need, the training can be provided, and we do training in a number of countries under OIE umbrella. Train their laboratories on disease diagnostics, and mainly for us it's classical swine fever, but the avian diseases unit does on avian influenza and foot and mouth disease. The vesicular disease unit at the NCFAD does some twinning projects in Nigeria. So we provide those help, and also we learn from for NCFAD. For us, these are all new diseases, and those countries who go through these diseases know better, and we don't know much about these things, and we learn as we provide the services for the diagnostics that we have developed to these countries. We learn as we go. Just before I close, I just wanted to I know we talk about all bad things, negative things, but you can eradicate these diseases, and we have done. And this is the first animal disease that has been eradicated from the globe. Rinderpest has been eradicated, but we haven't forgotten the damage it caused in the world for the cattle producers. The disease has been eradicated. It tells us that these diseases can be eradicated if we all work together. And I think next one is FMD on the, and then especially countries like Sri Lanka, and people have eradicated these diseases. Both developed countries as well as developing countries have eradicated a number of transboundary diseases by using the current tools. It is doable. It is not impossible. You can do it, but you need to have a plan. Okay, before I stop, I want to acknowledge again the iPURSE organizing committee for the opportunity, and I want to thank Dr. Munasinghe, the Dean of the Faculty, for the invitation, and also everybody at the Faculty of Veterinary Medicine, my alma mater, my teachers, my colleagues, and also the section heads who provided me slides on avian diseases, vesicular diseases, and the lumpy skin disease. My colleagues at the NCFAD. With that, I'm going to stop. I just want to thank everybody for joining today, and sorry if I went overboard a little bit at the time, but I have my email address down there. If anybody has any question, you can reach to me, and this is my team at the NCFAD. Probably you will recognize the person in the center. That's Kalahari, our new research scientist who joined me in the unit a couple of years ago. So thank you very much, and I'm happy to answer questions during our question sessions at the after Dr. Kothalawala's presentation. Thank you. I'm going to stop sharing my presentation now.
Okay, thank you very much, Dr. Ambagala, for that very interesting and informative presentation. Well, our next speaker is Dr. Hemali Kothalawala. She has already connected with us. Good evening, Dr. Hemali. Warm welcome to the webinar. We are excited to have you with us today. And Dr. Hemali will be talking about social economic impact of transboundary animal diseases. Let me formally introduce Dr. Hemali Kothalawala, the current Director General of the Department of Animal Production and Health. Dr. Hemali was a student of Vishaka Vidyalaya, Colombo, and she obtained the Bachelor of Veterinary Science degree from the Faculty of Veterinary Medicine and Animal Science, University of Peradeniya in 1994. She started her professional career as a government veterinarian. In 2011, she was promoted to the post of livestock economist of the Department of Animal Production and Health. There, her responsibilities include livestock sector planning, monitoring, and policy recommendations. She obtained a master's degree in animal science from University of Peradeniya. She further specialized in development economics by obtaining another master's degree in 2006 in livestock economics from Indian Veterinary Research Institute. Dr. Hemali obtained her PhD from the Obihiro University of Agriculture and Veterinary Medicine, Japan. Her PhD thesis titled Economics, Epidemiology, Integrated Approach to Brucellosis Control in Cattle in Sri Lanka. On behalf of Department of Animal Production and Health, she has contributed to several policy papers for livestock sector development in Sri Lanka. She has published extensively in national and international high-impact factor journals on economic impact of animal diseases. She has contributed to the professional development of veterinarians by holding key positions such as Vice President of Sri Lanka Veterinary Association and as the President of State Veterinary Surgeons Association. Presently, she is a special grade officer in the Sri Lanka Animal Production and Health Service. And she is the current Director General of the Department of Animal Production and Health. As far as I am aware, she is the youngest to hold the prestigious position throughout the history of the Department of Animal Production and Health. More importantly, Amidst her busy schedule, she still contributes to the undergraduate and master's degree program of the Faculty of Veterinary Medicine and Animal Science, University of Peradeniya, as a visiting lecturer in livestock economics. With that introduction, let me invite Dr. Hemali to deliver her speech.
Thank you, Dr. Kalupahana. Good evening, good morning, and good afternoon, those who are there in different time zones. Thank you all for inviting me for the organizing committee.
Dr. Hemali.
Sorry?
Okay, right. Now we can hear you now.
Okay. Yes. Thank you. I would like to thank you for the organizing committee for inviting me for this session, especially the Dean of Faculty of Veterinary Medicine and Animal Science, Dr. Madhura Munasinghe, and also Dr. Kalupahana, who is the moderator here, and Dr. Sampath Lokugalapatti also. Yes, my title is Socioeconomic Impact of Transboundary Animal Diseases. I think Dr. Ambagala has talked about the diseases mainly and the overview of most of the transboundary animal diseases. So I am going to touch mainly first very briefly livestock sector in Sri Lanka, and also the strength of Department of Animal Production and Health in animal disease control in Sri Lanka, livestock and transboundary animal diseases, a little bit about the TEDs again, and the economic impact of transboundary animal diseases, and also impacts of TEDs and their control. Yes, if I am talking about the livestock sector, you may know the agriculture contribution is somewhere around 7.2 to the total GDP in Sri Lanka. The livestock contribution is somewhere around 1%. In rupees, it's 225.8 billion per year. So we have dairy, poultry, goat, and swine livestock subsectors in the country. If I'm talking about the dairy sector, we do 40% demand sufficient. Actually, we have only 40% of the requirement we do produce inside the country. Most of our farms are subsistence level. Still, there are 1 or 2 animals, smallholders, dairy sector. So, but now we are in the transformation era. The subsistence level farms into small and medium scale enterprises, SMEs, and also commercial level mega dairy farms. If I am talking about poultry sector, 100% demand sufficient. So we don't import poultry and poultry products to the country. We only import inputs for the poultry farming for the country. Yes, so this pie chart discussed about the contribution of different subsectors to total livestock GDP. Chicken meat is the highest. It's somewhere around 105 billion rupees per annum. And also the second one is eggs, 44 billion per annum. And milk, beef, and other meat types are there respectively. The total GDP contribution is around 225.8 rupees billion. We do have exports despite imports of milk and milk products for 60 billion rupees per annum. This slide discusses about how we have grown in livestock product consumption over the last 30 years. Or yes, 4 decades actually, 40 years. Chicken meat, in 1980, it was 100 grams per person per annum. But now it has increased up to 10 kilograms. It's actually 100-fold increase. If I'm talking about the laying sector, the eggs number consumption, It was in 1980, it was 32 eggs per person. It has increased up to somewhere around 125. And milk, 17 kilograms per person per year. It has increased up to 57 kilograms per person per year. So this slide shows with increasing income, urbanization, and changing different socio-economics of the people, the livestock product consumption has significantly changed over past 40 years. This is about dairy farming in Sri Lanka. Dairy farming has been a traditional practice which has survived thousands of years in Sri Lanka. It is a secondary income generating source for many semi-urban and rural inhabitants. It's not mainly primary income generating source. Those who are doing agricultural farming mainly, this is done as a mixed crop, livestock and crop mixed farming system. Family labor involvement is high in the smallholder system. Low productivity, low marketing surplus, and high cost of production are main features of dairy industry. The productivity is so low, actually per animal, if I'm talking about average productivity per animal, and marketing surplus is also low in most of the systems. Production cost is high. Because of these reasons, the profitability is low. Animal diseases are one of the main reasons for low productivity and profitability. So these three pictures, the first one is intensive smallholder farming system. And the second one is, in the middle one, is semi-intensive farm. And the bottom one, extensive farm. Extensive farms are present mainly in northern province, eastern province, and sometimes northwestern and north central provinces as well. Yes, so the Department of Animal Production and Health. The Department of Animal Production and Health is the state organization which is responsible for providing technical leadership to the livestock industry and its stakeholders in Sri Lanka. The DAPH, which is located in Peradeniya, the hill capital of Kandy, Sri Lanka, is being functioned under the Ministry of Agriculture with the direct responsibility of State Ministry of Livestock, Farm Promotion and Dairy and egg-related industries. So this is the very brief organizational structure of the department. So there are 4 additional Director Generals, technical additional Director Generals actually, ADG Animal Health, ADG Veterinary Research, ADG Program Development and International Affairs. These 3 ADGs are directly involved in disease control and related activities. And we have another ADG, ADG Livestock Development. Under ADG Livestock Development, we have animal breeding and human resource development divisions. So these ADG Animal Health, ADG Veterinary Research, and ADG Program Development and International Affairs, the divisions comes under these 3 ADGs, Animal Health Division, Veterinary Research Institute, Veterinary Regulatory Affairs, and Livestock Planning and Economics. So we have several specialties, principal scientists in the VRI, and also specialists for different fields. The epidemiologist is also there in the department. This is the organizational structure of the Veterinary Research Institute that contribute a lot to disease control and the related research in the field. So we have under the Director Veterinary Research, Animal Production Deputy Director and Animal Health Deputy Director, and also Technical Services Deputy Director. Under Animal Health Deputy Director, Bacteriology, Parasitology, Pathology, Virology, and Molecular Biology divisions are there. All these divisions directly do their research related to animal health. And under the Technical Services Division, the Veterinary Investigation Center, Vaccine Production Center are also involved in vaccine production and disease diagnosis for the disease control activities. Yes, these are the pictures of VRI, Veterinary Research Institute, those who are doing field monitoring programs and also laboratory testings. And we have an animal virus laboratory at Polgolla that also comes under the VRI. Polgolla produce vaccines for foot and mouth disease. Foot and mouth disease, one major disease we have in Sri Lanka. So we do produce local vaccine at the VRI, the Polgolla, and do vaccinations all over the Sri Lanka for in the control programs. And there are veterinary investigation centers. Actually, we apart from the Central Veterinary Investigation Center at VRI, we have peripheral 26 veterinary investigation centers. So these are certain pictures of peripheral veterinary investigation centers. So in the peripheral veterinary investigation centers, they do supply services for the farmers, and also monitoring and surveillance programs are being done in these veterinary investigation centers. Yes, and I will move to the livestock pets and livestock sector. Livestock sector is one of the fastest growing segments of the agricultural economy, particularly in the developing world. It plays a key role in the livelihood of rural poor, contributes major proportion of their cash income, capital assets, draft power, fuel, and fertilizer. Increasing demand for animal origin food due to livestock revolution would provide income growth opportunities for poor. But low productivity, as I told earlier, common to Sri Lanka also, the quality due to livestock diseases are main issues. It's a global issue. Livestock diseases and poverty are closely related all over the world. So the transboundary animal diseases, the topic today, you know now, of significant economic, trade, and or food security importance for a considerable number of countries, and neglected zoonosis that affect human and animals. For example, brucellosis, tuberculosis, rabies are embedded in developing countries. So transboundary animal diseases are embedded in developing countries, including Sri Lanka. Global, if I'm talking about global transboundary animal diseases, pets, in mid-1990s, the term transboundary animal disease, TED, has become widely used for a group of major infectious diseases of livestock with important economic impact. The globalization, as Dr. Ambagala mentioned, the globalization of food and feed trade, facilitated by liberalization of world trade, while offering many benefits and opportunities, also represent new risk. And food, a major trade commodity, is also an important vehicle for transmission of infectious diseases. Dr. Ambagala also discussed these points, because food production, manufacturing, and marketing are now global. Infectious agents can be disseminated from the original point of processing and packaging to locations thousands of kilometers away. That's why we do have transboundary animal diseases now. Yes. So why the stats are there? Globalization and international transport. Dr. Ambagala also discussed these points. Environmental changes, ecological interface, husbandry and technological changes, spread of animal farming into new ecosystems, and also break in animal health service and infrastructure. Actually, these are the causes of threats in globally. Yes. So if I am talking about the impacts of diseases, scientifically, there are different categories of impacts. Disease impact can be divided into main two groups, direct impact and indirect impact. Direct impact are visible losses and sometimes invisible losses. Visible losses that we can see, reproduction or production loss, loss of draft power, loss of weight gains, dead animals. These things we can see, but there are things we can't see. Low fertility, change in the herd or flock, we can't see. Yes. And also indirect impacts are there. Additional cost, yes, it's indirect impact. Vaccines, vaccination, vaccine delivery, movement control, diagnostic test, research, extension, infrastructure development. All these things are additional cost because of diseases. And also social cost is there. Trade disruption, denied access to markets, and destruction of rural economy are social impacts because of diseases. So these are the overall picture of disease impacts of these transboundary animal diseases. Yes, so this shows the relationship between the losses and expenditure, losses and control cost actually. So when the cost on the control is low, the losses are high. And when the cost on control is high, the losses are low. This is how the relationship behave, losses and expenditure in between related to diseases. Yes, the cost benefit analysis is one tool commonly used to study about the control strategies and also the impacts of diseases. The cost benefit analysis on foot and mouth control program for 2005 to 2010 in Philippines have studied. So it says the benefit cost ratio or the BC ratio was 1.6 with no exports. But when they include the exports, it has increased up to 12, the BC ratio, when included the exports. So this shows how the benefits are increased when you consider the trading part in your cost benefit analysis. Losses are higher than control cost always because of that. And also another scientific studies are there. This is one of that. The economic impact of foot and mouth disease and its control in Southeast Asia, a preliminary assessment with special reference to Thailand. So in that study, it says, if FMD were to be eradicated from Thailand in 2010, the eradication would be economically viable, even without exports, with a predicted benefit cost ratio of 3.73. With additional export, the economic justification for control becomes much stronger, with a benefit cost ratio up to 15.1 being achieved. So we can see how it increases when we consider our indirect cost segment. If eradication is not achieved until 2020, returns remain positive without exports, but a lower rate. This is another study about economic impact of lumpy skin disease and cost effectiveness of vaccination for control of outbreaks in Ethiopia. In that study, it was recorded 94% of herd owners rank LSD as a big or very big problem for cattle production. A large proportion, 92.2% of the herd owners indicated that LSD affects cattle marketing. Median loss of $375 in local Zebu and $1,250 in Holstein Friesian local Zebu cross cattle was estimated per dead animal. The median losses per affected lactating cow were $141. And also the diagnosis and medication cost per affected animal were estimated at $5. The median total economic loss of an LSD outbreak at the herd level was US dollars 1,176 according to that study. So this shows how important or what is the economic impact of these diseases. The socio-economic impact of avian influenza outbreak on small-scale producers in Indonesia says the number of farms stopping operations were 30%, and in the high incidence area, nearly 70%. The proportion of income from poultry for daily household expenditure has decreased from 75% to 91% before to 38% to 82% after the HPI outbreak. Direct impact of HPI was also seen by decrease in expenditure for education and daily consumption, in particular in the high incident farms. Yes, and this is also another finding of cost of AI in Southeast Asia, Cambodia, China, Indonesia, Laos, Thailand, and Vietnam. So it was estimated the cost for the population per person per day, and the percentage of rural and the percentage of labor in agriculture. Cost incurred in strategic planning, compartmentalization, early warning, vaccination, farm biosecurity measures, and market biosecurity measures also counted in this study. Yes. So the world is working on to control transboundary animal diseases. So many publications and work are being done on transboundary animal diseases. This is one is in SAARC countries, and there are reviews and studies are going on transboundary animal diseases and impacts on international trade mainly. Yes, as Dr. Ambagala mentioned, the GFTEDs, the Global Framework for Progressive Control of TEDs. So OIE and FAO has set a framework globally to control these diseases as a progressive control path. So in these studies, so objectives of these TEDs actually, the country-based surveillance and enhanced disease reporting, establishment of regional support units, the regional and global early warning systems, internationally verified global freedom from rinderpest, and animal populations where primary endemic circulation of FMD and other selected TEDs are identified and characterized. International, regional, and national early response capacities for prompt and authoritative disease diagnosis, referral diagnostic and molecular biological capacity of OIE, FAO reference laboratories, assistance in development of TED research. So GFTED, so the global framework includes these activities for all the countries, member countries. Yes, then I'll move on to the TEDs in Sri Lanka, what we do have in Sri Lanka. Yes, FMD, foot and mouth disease. Sri Lanka is endemic for FMD. So we do get cases or outbreaks annually for foot and mouth disease. And recently we had lumpy skin disease, LSD. Yes. And also PPR, Pestides petis in goats, CPD, contagious pustular dermatitis, and African swine fever. We have not yet, it's not reported in Sri Lanka. But we had classical swine fever earlier, but now the reported cases are not in recent past. Rhinicat or ND, Newcastle disease in chicken, is present in Sri Lanka. And also HPAI, highly pathogenic avian influenza, we do not have, so it's not reported yet. So the diseases in blue color are present in Sri Lanka, and others actually luckily we don't have. Yes, economic impact of the different diseases I'm going to talk. FMD. FMD is endemic in the dry zone of Sri Lanka. Over 70% of low country dry zone is earning living through farming. Around 60% of the farms are there in the dry zone of Sri Lanka. Average milk production is low, but it contributes to the national beef production. The FMD has recorded significantly and shows significant losses on milk production and meat production in the dry zone in Sri Lanka. Yes, survey has been done in 2008. According to that survey, the infections in extensively managed herds in the dry zones were high. In those herds, 50% of milking animals were infected, 10% of the pregnant dry herd, and 20% of the heifers and 30% of the calves have been infected. And calf deaths were there, 20% of extensive system and 4% in semi-intensive system farms had calf deaths. The milk production loss was somewhere around 41.4 in extensive system, even after 3 months of infection. So in foot and mouth disease, even after the 3 months or maybe more, still the milk production loss is there. So impacts of FMD is calculated: milk loss, calf deaths, meat loss, loss on delaying AIs, labor cost, treatment cost. It has been total to 16.3 rupees million for the dry zone of Sri Lanka. Yes. So impacts of FMD in Sri Lanka: development destruction. If any area is infected with FMD, the area is notified as an infected area. Movement restrictions are imposed. So all the veterinary services, extension services, and development activities should be temporarily ceased until the area gets cleared, according to the Animal Disease Act, number 59 of 1992. And also the trade disruption is there. Local milk collection, milk processing, milk price increase of human goods are being disrupted because of foot and mouth. And also the international trade disruption is there. Yes, this is the progressive control pathway for foot and mouth. Yes. So we are in the stage 2 of this pathway. So then now our next step is stage 3. So we have to jump to stage 3 from stage 2. According to the stage 2, the control program is implemented in risk areas in Sri Lanka, especially in the dry zone. And also we do produce vaccines and we do vaccinate these animals. We have a good control program. So because of that, Sri Lanka is in stage 2. And we are planning to step to the next step recently. Yes, these are the FMD control, pictures of FMD control programs at field level, vaccination programs. Yes. Then the economics of lumpy skin disease in Sri Lanka. We got LSD in 2019, first time. So the milk production loss was there and beef production loss was there. Weight loss and reproduction loss was there. Hide rejection was there because of LSD. So we had diagnostic cost and control cost or vaccination cost because of lumpy skin disease. Economic impact of swine diseases in Sri Lanka. We had, as I said earlier, CSF, classical swine fever. Outbreak was there in 1998 and also PRRS in 1999 and 2019. High mortality is there. So income disruption was there for farmers. So the government has to incur the diagnostic cost and the control cost for vaccination for these diseases. Economic impact of poultry diseases. Ranicot outbreak was there in 2010. Over 100,000 birds mortality was there. The estimated loss was 60 million rupees. And each year, sporadic outbreaks are there, but not as 2010. So we do incur diagnostic cost, control cost for for regular vaccinations. VRI does produce local vaccine, and also we have imported vaccine. And we have extension programs, so we have to incur cost for extension programs as well because of pandemic. Yes, and also we have to incur certain cost for diseases which is not present in Sri Lanka. So to prevent entry of these diseases, the quarantine measures, pre-quarantine and post-quarantine measures are there. So we do have quarantine stations. Katunayaka Airport, we do have a quarantine station, and also for Seaport, we had quarantine station at Morgan Road, Colombo, and we do have a quarantine station at Mattala Airport. And also recently, we established one in Jaffna as well. So the quarantine, we have to maintain our quarantine measures to stop entry of transboundary animal diseases into the country. And also regular surveillance are going on. Disease investigations are going on through our disease investigation centers and the Veterinary Research Institute. The disease reporting system is there, and the extension on biosecurity measures are being done. And also we have preparedness plans for HPAI and ASF. What we are going to do if we get these diseases. So we have preparedness plans for these two diseases. So we are empowered with, as the department, we are empowered with legal provisions of Animal Disease Act of 59 of 1992 and Animals Act of 29 of 1958. Animal Feed Act number 15, disease control, and also work with the quarantine stations and all. Yes, and I'm going to talk on sanitary and phytosanitary standards. Actually, sanitary and phytosanitary measures play a role in high priority in international trade and access to new exports markets. SPS measures which confirm the international standards, it's international standards actually, guidelines or recommendations shall be deemed to be necessary to protect human, animal, and plants. SPS stand for sanitary and phytosanitary standards. Impact of some zoonoses and foodborne disease outbreaks or plant life or health and presumed to be consistent with the relevant provisions of agreement, SPS agreement. So SPS agreement is there. So to maintain the consistent all over the world, so SPS agreement is there. So SPS agreement is according to the World Trade Organization's WTO agreement. So agreement on this application of sanitary and phytosanitary measures, the SPS agreement, entered into force with the establishment of WTO on 1st of January 1995. It allows countries to set their own standards, but also says regulations must be based on science. They should be applied only to the extent necessary to protect human, animal, and plant life or health. And they should not arbitrarily and unjustifiably discriminate between countries where identical or similar conditions prevail. Also, in SPS agreement, there are 14 articles on general provisions, basic rights, harmonization, equivalence, so on, and also 3 annexures, definitions, and annexure B, and transparency of sanitary and phytosanitary regulations, annexure 3, control, inspection, and approval procedures. So SPS agreement give a guideline or international standards to protect entering transboundary animal diseases into countries. Yes. So the standard setting organizations are FAO, WHO for Codex Alimentarius Commission, Codex for Food Safety, and World Organization of Animal Health, OIE for Animal Health, FAO, International Plant Protection Convention, IPPC for Plant Protection. So these are the standard setting organizations for above SPS measures. Yes. So these organizations OIE, or World Organization for Animal Health, World Trade Organization, World Health Organization, FAO, Food and Agriculture Organization, and also IPPC, International Plant Protection Convention. All these agencies work together to prevent ticks across the world. Thank you.
Okay, thank you, Dr. Hemali, for that very informative and interesting presentation again.
Yes, if you have any questions, I can answer.
I have to ask few
Okay,
questions.
Yes.
Let me check the chat. Okay, so as there are no questions there, I want to pose some questions for the presenters. The first one is for Dr. Ambagala. So Dr. Ambagala, so few years ago when the HPI came into the South and Southeast Asian countries, there were a lot of pandemic preparedness plans and a lot of programs and training programs. And ultimately, nowadays, the concentration is on corona, right? Coronaviruses. Yes. And most of even some of the scientists, I have discussion with them, they feel that now HPAI is almost like a spent force. It's not relevant or not having the potential to create a pandemic. So as a trained molecular virologist, I want to ask from Dr. Ambagala about his opinion about this HPAI condition, high pathogenic avian influenza. Dr. Ambagala?
Yes, can you hear me? Hello? Hello?
Yes, we can hear you.
Yeah, okay, you can hear me. Okay, so yeah, thanks, Anil, for the question. And yeah, so I mean, currently people are struggling with COVID-19. COVID-19 is the top priority, but that doesn't mean that other diseases are causing problems, right? Of course, we talk about the global trade, and global trade happens, and birds are moving, and so the threat from highly pathogenic influenza is not gone. It is not in the main on the top of the concerns, but it is a concern, and many HPAI outbreaks have happened over the last couple of years. Currently, Ghana we just identified a high pathogenic avian influenza virus in Ghana. So, and the possible of human exposure. So these things happen. So of course, the viruses mutate, and the rest of the things. So it is not gone. We will continue to have issues with highly pathogenic avian influenza after the COVID is gone. So it is a continuous threat that we will face.
Thank you very much for that answer. I have another question. Can you tell us how the LSD came to Sri Lanka? What is the most probable way that it has reached this part of the world? I know it is spreading to the South and Southeast Asian countries from Africa. Gradually, what is the reason behind that, or what is responsible for carrying this organism to this part of the world?
Yes, actually.
Yeah, I think Hemali can answer that question better than me.
Yes, Dr. Ambagala also can contribute. Yes. Yes, actually, I think though we have quarantine stations and we do follow quarantine measures, illegal, the smuggling is their animals. So we suspect it came through such kind of importation, smuggling actually. Because in the peninsula, sometimes such kind of smuggling is it's common. So we suspect actually, yes, that was the route to enter LSD into the country.
Yeah, I agree. I think the most likely route is illegal importation of animals, and some of these diseases that transmit mechanically through vectors, and the vectors can play a major role too. Like people have seen in the United States when it comes to vesicular stomatitis disease, where the flies are involved in transmission of the disease, and they can transmit pretty long distance, and it is even possible to transmit through the midges to the flies across the ocean, and that has happened, and that has been shown with other diseases. Even a mechanical transmission is possible through a long distance by the vectors, but in this case, I think it is, like Hemali pointed out, I think it is very likely illegal transport of animals, which is difficult to control, happens all over the place. And in United States, there was a illegal, they captured a boat full of pork meat, large amounts, I don't even remember, 50 tons or something, trying to enter into United States. So the illegal transport is pretty common, and so it's very likely that that's the route.
Okay. Thank you, Dr. Ambagala and Dr. Hemali, for that answer. And I want to ask another question, if we are not constrained with time.
Yes,
so
yes, Dr.
I want to ask especially from Dr. Hemali, is there any plans to improve diagnostic and control services, disease diagnosis and control services by the Department of Animal Production and Health, and as the university, what kind of contribution we can give to that to make it more effective?
Yes. Actually, yes. Thank you for that question, Dr. Kalupahana. Yes, actually, we are in the process of developing or strengthening our veterinary investigation, peripheral veterinary investigation centers to decentralize certain services, and also the center, the VRI, Veterinary Research Institute. Actually, I think now the faculty has good laboratories with faculty, so we can do the collaborative research as well as the diagnosis, and the present also in certain diagnostic facilities, providing diagnostic facilities for the department, because we do not still have COVID-19 test at the VRI. So for imported animals, so the faculty provide that service. So we can strengthen our links and do a better job for diagnosis and surveillance and monitoring, whatever it is, to control these diseases, faculty and the VRI and the DAPH. And also, I think Dr. Ambagala also can help us, with your knowledge, as well as some other, if you can, some other facilities. So I take this opportunity to make that bridges in between the faculty and also with Dr. Ambagala's laboratory and with the department. Thank you.
Thank you, Dr. Hemali. There's another question from the audience. Professor Anil Pushpakumar is asking, smuggling of live animals pose a real threat for the introduction of new diseases to Sri Lanka.
Exactly.
So what is the plan to tackle this issue?
Yes, actually, we are in the process of having discussions with the Navy and the Northern Peninsula. And also now we have a officer at the Jaffna airport. Very recently, we established that facility. So we have the collaborations with this Navy personnel and our veterinary, the quarantine officers and the veterinary investigation center. We are going to have a program to monitor, but it is not very easy thing. not that easy, but still we have a plan to have a collaborative program with them.
There are no other questions from the audience. Let me thank Dr. Aruna Ambagala and Dr. Hemali Kothalawala for very interesting presentations today. Now we have to wrap up the session and it is well past 7 o'clock. Let me formally thank once again to Dr. Ambagala and Hemali Kothalawala for sharing valuable and insightful information with us. And I will hand over the control to Dr. Sampath Lopugalapatti to wrap up this webinar.
Okay, thank you very much, Dr. Kalupahana, for moderating the session, and I would like to thank both our resource persons, Dr. Hemali Kothalawala and Dr. Ambagala, for sharing their very informative and very insightful speeches with us. And it was very, very productive in the sense that we were made to knowledgeable on the latest situation of these transboundary animal diseases and how we can tackle them, how we can prepare in case of in an outbreak, how we can prepare for a response in a phase of an outbreak. So both for resource persons and particularly for Dr. Ambagala sharing the global situation and for Dr. Hemali Kothalawala for enlightening us on the national context and what plans we have. So I think that's very important for all of us as veterinarians to think how we should manage them, because according to the statistics shown by Dr. Hemali, the economic losses are huge for the country. So it's a global scenario, so that I think all the information provided are really valuable for us to plan ahead and prepare for the to tackle the transboundary animal diseases. So let me formally thanks once again for both resource persons. And I would like to thank our moderator, Dr. Kalupahana. And also I would like to thank all the logistics supports provided by the iPURSE Secretariat, as well as the International Relations Office of the University of Peradeniya. And I would like to thank all the participants who joined from locally as well as from the North America particularly. Let me conclude the sessions and adjourn the sessions by wishing you a very pleasant evening. Thank you very much.